Previously, we discussed about glycogen and metabolism, including glycogen synthesis and glycogen breakdown. In this session, we'll talk about how these two pathways, that is glycogen synthesis and degradation, they are regulated. The objectives of this session are, we'll study general aspects and types of regulatory mechanisms, and the second objective is regarding allosteric regulation. Before starting, why regulation of metabolic pathways? Why any metabolic pathway shall be regulated? A metabolic pathway which is taking place in the cell has to be regulated. If well-regulated pathway means it is a normal condition. If the pathway is not properly regulated, it can be treated as abnormal condition. Okay, that may be related to the carbohydrates, that may be related to the proteins, that may be related to the fat and lipids. Let us talk about glycogen regulation. Why glycogen has to be regulated? Why glycogen shall be regulated? From the glycogen, glucose is released. Glucose is released. The glucose is required, it goes to the blood, and so how much glucose is going on, okay, how much glucose is going to the blood, okay, so that has to be regulated, maintaining of, so blood glucose, okay, so we cannot allow excess glucose to enter the blood. Also, very low glucose entering the blood, it has to be properly maintained and balanced depending upon whether the person is in a fed state or fasting state. As I said, glycogen, there is a synthetic pathway and also there is a degradative pathway, degradation and synthesis. So these two pathways are tightly regulated. While dealing about this pathway, when I said the pathways are completely different pathways using different set of enzymes. It is not reversal of same pathway. There are completely two different set of enzymes are there in the glycogen synthesis as well as glycogen degradation. Let us talk about fed state. What is the situation in fed state after taking the meals? Okay, then the fed state, so there is plenty of glucose is available. When the plenty of glucose is available, the glucose shall be directed to the storage in the form of glycogen. So the synthesis is increased. In the fed state, there is no need of the degradation of glycogen. There is no need of supply of glycogen by glucose by breaking it. This is the scenario in the fed state. Let us talk about the fasting state. Fasting state, that is opposite to the fed state. The person is in a fasting state where glucose is not available from the dietary sources. In that condition, what happens? The synthesis of glycogen should be stopped or almost should be stopped or decreased. And whereas glucose has to be supplied in the body by the cells, so the degradation pathway has to be increased. That means in the fed state, the synthetic pathway shall be increased. In the fasting state, the degradative pathway shall be increased. That is the reason. The regulatory mechanisms, so they are very important. If one pathway is active, another pathway shall be inactive. Okay, what are the different regulatory mechanisms? The regulatory mechanisms majorly two. Okay, so two types of regulatory mechanisms are there. So one is allosteric. One, it is, we call it allosteric regulation. The other one is hormonal control. Through the hormonal control, the regulation is taking place. The next aspect is, in this session, we will concentrate on allosteric regulation. we will concentrate on allosteric regulation. So in the allosteric regulation, again, I am going into the one first thing is well-fed state. What is the situation in the well-fed state? So let us see in other perspective, which is different from what we discussed just now. Let us see. In the well-fed state, there is high level of glucose. There is high level of glucose, then high level of glucose 6-phosphate, and also there is high level of ATP, high supply of ATP. So these three are available. So in this condition, so what happens here? In this condition, it happens, suppose when I talk about the glucose 6-phosphate and also I talk about the ATP and glucose, other things I will discuss later, glucose 6-phosphate and ATP. So what happens here, these two, when there is increased one, then they act as a positive modulator. So what do you mean allosteric modulators? The allosteric modulators we have studied in the enzymes. There are enzymes which are called as allosteric enzymes. Those enzymes which have a site other than the catalytic site. This site we call it allosteric site, where these modulators bind it. By binding this modulator, the enzyme activity either will increase or decrease. If enzyme activity increases, we call it positive modulators, and if it is decreases, we call it negative modulator. Okay, let us see in this case, glycogen synthase and glycogen phosphorylase in the well-fed state. So, in the glycogen synthase, in the well-fed state, glucose 6-phosphate increased, ATP is increased, so they act as a positive modulator, leading to the increased glycogen synthesis, leads to increased synthesis of glycogen. Synthesis of glycogen, that is the because of positive modulator. Whereas the same time, these glucose 6-phosphate and ATP, they act as a negative modulator. So decrease glycogen phosphorylase activity that leads to the decreased or stoppage of glycogen degradation. See, the modulators, if they are positive, they increase the activity, and if it is negative, they decrease the activity. Let us talk about the fasting state. In the fasting state, it is just reverse. In the fasting state, it is just reverse. So there is decreased glucose 6-phosphate and there is decreased ATP. Of course, there is a decreased glucose. Okay. Then here it is the glycogen phosphorylase will increased activity of glycogen phosphorylase. Okay. So leads to increased degradation. It is increased degradation. Okay. So I will explain how it is in the fasting state. Glucose 6-phosphate is decreased, ATP is decreased. In that condition, what will happen? The positive modulator effect will not be there on the glycogen synthase. In that condition, the enzyme is not active. And simultaneously, when there is a decreased glucose 6-phosphate ATP, the negative effect of these allosteric modulator will not be there on glycogen phosphorylase. So in this condition, the glycogen phosphorylase will be active in the absence of this negative modulator. This is about the allosteric effect of this product or one of the intermediate of this pathway, glucose 6-phosphate and also ATP. Okay. Then the next one is a calcium plays very important role. The calcium 2 plus plays very important role, especially in the activation of degradation. Okay, so in the degradative pathway, calcium plays very important role. Okay, what happens during muscle contraction? During muscle contraction, more and more ATP is required. Then there is increased need for ATP. From where this ATP comes? This ATP is supplied from glycogen. Okay, this ATP is supplied from the glycogen. Okay, let us see the mechanism here. Nerve impulse. So nerve impulse, what happens? In the nerve impulse, in a biochemical aspect, here there is increased membrane polarization. Membrane polarization. Okay, when nerve impulse takes place, then increased membrane polarization is taking place. Then that will lead to the increased release of calcium 2 plus. Calcium 2 plus release is increased. Calcium 2 plus release is increased from where? From sarcoplasmic reticulum. Sarcoplasmic reticulum, the increased release of calcium is taking place. Okay, then am I clear? Okay, once this calcium is released, let us see, we have calcium 2 plus. Then what further takes place? Let us see here. So there is a protein, that is, it is specific for calcium. The protein is specific for calcium. Protein is called calmodulin. The protein is called calmodulin. Okay. The calmodulin protein, what happens here? Same, in the presence of the calcium, what happens? Here again I am writing. Okay. So what happens? The binding of calcium is taking place. Here, this is one calcium, the second molecule of calcium, ion of calcium. This is the calcium which is binding. This is, we say, this is calcium. Okay, here, this is calcium 2 plus. Okay, then what we say is calmodulin, okay, calmodulin plus calcium complex. Calmodulin, calcium complex. Okay. So once this one binds it, what happens? Here, this binding protein with the calcium, what it does, it will bind with particular enzyme. This is the enzyme. This is the enzyme. So the enzyme, when it binds it, it becomes active enzyme. Okay, once it is active enzyme, then what will happen? The substrate is converted to product. Substrate is converted to product. Okay, that is what happens. Let us take this name of the enzyme, in this case, phosphorylase kinase. The phosphorylase kinase is very crucial enzyme in the regulation of glycogen degradation. Okay. When you talk about this calcium calmodulin complex, the calcium plus the calmodulin, the protein, calcium binding protein, okay, it can act independently. It can act independently. Independently means what? So, it is independently of cyclic AMP controlled regulation. Cyclic AMP related protein kinase that we'll study in hormonal regulation. Without the cyclic AMP dependent protein kinase, this calcium 2 plus, so calmodulin complex, can act independently in the regulation of glycogen degradation. Okay, that is the one more aspect. The next one, I'll talk about AMP. Adenosine monophosphate, not cyclic AMP. It is also act as allosteric modulator. It also can act as allosteric modulator. Suppose in the muscles, this particular AMP, how it acts. Here in the extreme conditions, there is very low levels of ATP. In that condition, what happens? Here, there is very high increase of AMP. there is high increase of AMP. Once there is high increase of AMP, what will be the effect of this AMP? What it does, inact to phosphorylase. This will act on this phosphorylase enzyme, which is main enzyme of glycogen metabolism, to act to phosphorylase. Once the phosphorylase is active, then so what happens? The glycogen degradation will increase, leads to increased degradation. Okay, so that is AMP as modulator. One thing you will come to know later, phosphorylase will be active if phosphate is added. But here, without the addition of the phosphate, this enzyme becomes active. Okay, enzyme becomes active. So due to the effect of AMP. Okay, so this is about the allosteric mechanism. I will summarize today's session. We have taken up regulation of glycogen metabolism. The objectives we set in this session are general aspects and types of regulatory mechanism. Then we focused on the allosteric regulation. Then we said why regulation. We said regulation is a must process to keep in a cell in normal state. If a particular metabolism is not regulated, that is not a normal condition, that can be considered as abnormal condition. We are talking about glycogen regulation, both synthetic pathway and degradative pathway. So glycogen, why? There shall be regulation. Glycogen is responsible for the release of glucose. Glucose is released into the blood. The glucose has to be well controlled. Release of glucose into the blood should be there. So that is the reason the synthetic pathway and degradative pathway shall be tightly regulated. This is very essential. And we also talked about fed state. What happens? In the fed state, synthesis will increase as plenty of glucose is available. Degradation pathway is decreased. In fasting state, it is opposite. The synthesis is decreased. Degradation is increased as there is a lack of glucose supply. Okay, the dietary supply. Regulatory mechanisms, they're mainly allosteric regulatory mechanism and hormonal regulatory mechanisms. In the allosteric regulatory metabolism, again we took up the well-fed state where we said glucose levels are increased and glucose 6-phosphate they are increased, ATP are increased. The glucose 6-phosphate and ATP, they act as this glucose I will discuss later in a separate way. The glucose 6-phosphate and ATP, so they act as a positive allosteric modulators, increasing the glycogen synthase and synthesis of glycogen. So we also talked about the allosteric enzymes. There is a site other than the catalytic site where these modulators they bind. Either they will show the positive effect or negative effect. If positive effect is shown, we say positive modulators. If the negative effect is shown, they are called negative modulators. Here in this case, they are the positive modulators. Here, these when they bind, so they decrease the glycogen phosphorylase activity. They are negative modulators. Degradation of glycogen is decreased. And okay, degradation of glycogen is decreased. Okay, then here we have. Then what happens? In the fasting state, these levels are very low. In this condition, the positive modulator effect will not be there and glycogen synthase, the glycogen synthase activity is decreased. Glycogen synthase activity is decreased. Then what happens? Glycogen synthesis is decreased. And that negative modulator effect will not be there because of very low glucose 6-phosphate ATP and the glycogen phosphorylase activity will increase. Glycogen phosphorylase activity increase and degradation will increase. Okay, then we also talked about calcium. Calcium in the activation, okay, in the activation of degradative pathway. Okay, then during muscle contraction, there will be need for the high ATP that is supplied from the glycogen, okay, by degradative pathway. So when the nerve impulse, what happens? There is a membrane polarization increases that leads to the increased calcium 2 plus release from sarcoplasmic reticulum. Okay, then the calcium 2 plus so binds a protein called calmodulin, and the calmodulin four calcium ions, they can bind it and leads to calcium calmodulin. The complex is formed. When inactive enzyme binds it, that becomes the active enzyme. The active enzyme converts the substrate into the product. The example which we have taken, phosphorylase kinase, very crucial enzyme in the regulation of degradative pathway. Then this calcium calmodulin can act independently of cyclic AMP mechanism of regulation that we'll study in the hormonal section. Then we also talked about AMP as allosteric modulator in the muscles. Then extreme conditions in anoxia and also ATP depletion. Okay, in anoxia and ATP depletion, so lack of oxygen here, lack of oxygen, and then in the also in the ATP depletion. So what happens? Fatigue of the muscles takes place. Then ATP, the increased AMP, so will be there. The increased AMP. Okay, increased AMP causes the inactive phosphorylase to the active phosphorylase. And okay, so then increased degradation. So without this takes place without the addition of phosphate, without the addition of the phosphate to the phosphorylase, to the phosphorylase. So that is phosphorylase. Also that we'll study later. So that it can be active once the phosphate is added. That is in the hormonal regulation that we'll take up in the next session. Okay, thank you.